We're going to talk about organic compounds. Organic compounds always contain the elements carbon and hydrogen. Many organic molecules are soluble in water. We're going to start by talking about carbohydrates. The carbohydrates are the most important source of energy in our body. Carbohydrates are organic molecules that contain carbon, hydrogen, and oxygen in a ratio of 1 to 2 to 1. So for every 1 carbon, there's 2 hydrogens and 1 oxygen. Carbohydrates only account for about 1% of total body weight. Sometimes the carbohydrates can be very simple carbs, and we would call those monosaccharides. They are simple sugars containing only 3 to 7 carbon atoms. So here we see a monosaccharide that has 1, 2, 3, 4, 5, 6 carbons, and it's more in a straight chain form. Over here we see a very popular form of carbohydrate, which is glucose, and glucose is in this ring formation. And so we see all these different carbons, 1, 2, 3, 4, 5 carbons, and then we can see that there's this functional group off to the side with a 6th carbon in it. Down here we see a 3-dimensional model that shows the organization of atoms in the ring. Here we're seeing a little larger carbohydrate that's called a disaccharide. The disaccharide is made up of 2 monosaccharides. Di means 2. So a glucose and a fructose molecule. Now glucose and fructose are called isomers, and what that means is that they have the same chemical formation. It's just that it's arranged a little bit differently. So they have the same number of carbons, hydrogens, and oxygens. They're just all arranged a little bit differently. And what we're seeing here is a reaction that is pretty common, and we'll hear of this throughout the semester. It's called dehydration synthesis. Dehydration implies that water will be removed, and water is H2O. Synthesis implies that we're going to be making something bigger. So we're going to take 2 smaller molecules, combine them together to make something bigger, and that's exactly what we have here. The glucose molecule and the fructose molecule are going to bind together by removing water. So we can see that 1 hydrogen is removed from the fructose, and a hydroxide, or an OH, is removed from the glucose. When that is removed, we end up with water, and the oxygen that's left over will bond to the opposite carbon in the glucose molecule, and here we have that bond right here, and we end up with a compound called sucrose. And so that is a disaccharide. It contains 2 different carbohydrates. Now we can do the exact opposite by adding water, and adding water, we call that hydrolysis. So here's our H2O, and the water will be added to the compound sucrose, which is a disaccharide, and when water is added, the hydroxide will go to one of the rings, and the hydrogen will bond to the other ring to give us both a glucose and a fructose molecule, and so now we're back down to monosaccharides. Okay, so a hydrolysis is the process of adding water to break apart a larger molecule into smaller molecules. Carbohydrates can be stored in large molecules, which would then be called polysaccharides. Poly for many, saccharides for the sugar or the carbohydrates. And they can be stored as either starch or glycogen. And glycogen is stored in the liver and in the skeletal muscles. So there are organelles in the liver and in the skeletal muscle that will take a single glucose and through dehydration synthesis bind it to another one, and then to another one, and then to another one, until we get this whole string of carbohydrates, this whole chain. And then we would call that a polysaccharide, and this particular polysaccharide is glycogen. You'll hear that word over and over and over again. It's just a way that we store glucose molecules. Here is another picture depicting what glycogen looks like. It's just chains of glucose molecules. Now we're going to change to another organic molecule that's called a lipid. Lipids account for 12 to 18% of the total body weight of the adult male and 18 to 24% of the total body weight of the adult female. So a little bit more of the weight than the carbohydrates did. Fats have both fatty acid chains and glycerol. Here we're seeing a chain of fatty acids, and we're also seeing a glycerol, and together they're combining to form this lauric acid. Now the fatty acids are long chains of carbons, and so here we see carbon, carbon, carbon. All these long chains here are forming the fatty acid, and so we're also seeing them here. Okay, long chains of carbon atoms that are joined together. And then at one end we have the glycerol group. There are two different types of fatty acids. There's either a saturated fatty acid or an unsaturated fatty acid. The saturated fatty acid is a chain of carbon atoms with single covalent bonds. So there's only one single bond between the carbon atoms. In the unsaturated fatty acid, there's going to be at least one carbon-to-carbon bond that's a double bond. When a glycerol group is bonded to a fatty acid, that would be called a glyceride. Through the process of dehydration synthesis, a fatty acid could bond to a glycerol group to become something larger. And so when there's only one glycerol group and a chain of fatty acids, we would then call that a monoglyceride. If two fatty acid chains are bound to a glycerol group, then it would be called a diglyceride. And if three fatty acid chains are bound to the glycerol group, that would be called a triglyceride. Triglycerides are important. They give us energy, so they can be used as an energy source. They are stored in the fat cells, which give us insulation as well as protection. Another type of lipid is called a steroid, and steroids can come in the form of cholesterol or in the form of hormones that we're familiar with, estrogen and testosterone. We can see that they are distinctly different. They don't have the glycerol group with the fatty acid chains. Instead, we see that they have a distinctive carbon framework with 4 rings of 5 to 6 carbons that are attached to various side chains. So here we have 1, 2, 3, 4 rings, each of those having 5 to 6 carbons, and they are attached to various side chains. Even estrogen has 1, 2, 3, 4 rings. Testosterone, 1, 2, 3, 4 rings. Cholesterol is important because it helps to maintain the cell membrane. Cholesterol also helps to regulate metabolism in the tissues. It also helps to form bile salts. Bile salts help to break down fats. Estrogen and testosterone help to regulate sexual function. Cholesterol is derived from animal products. We have to eat animal products in order to get the cholesterol, and they are also synthesized in the liver. So we also have a little genetic component on how much the liver will synthesize. The last 2 lipids that we want to talk about are called phospholipids and glycolipids. The phospholipids are found in the cell membrane, so we're going to talk a lot about that in Chapter 3. The phospholipids have a head which contains a glycerol group as well as a phosphate group and then also a non-lipid group. So it's a pretty big head. And then there are 2 fatty acid chains or 2 fatty acid tails coming off of the head. The head is going to be what we call hydrophilic. And what that means is that it's water-loving. And so the hydrophilic head will be closest to the water, either closest to the inside of the cell or closest to the water in the outside of the cell. The 2 tails are going to be hydrophobic. Hydrophobic means that they are water-hating, water-fearing, and they will actually repel from the water. And so when we see them in a bilayer, they will be facing each other, and we'll see that when we get to Chapter 3. The glycolipid is a carbohydrate that's attached to a diglyceride. We see a carbohydrate, and there we can see with the 3 rings. We know that that's a carbohydrate because of the rings that you're seeing, and then also the 2 fatty acid tails that are called a diglyceride. When placed in water, these phospholipids are going to form a mycel. Over here we can see what a mycel looks like. Again, the heads are hydrophilic, so they're going to point towards the outside because there's water outside. And then when you look inside, you'll be able to see all the tails that are facing in towards each other because they are hydrophobic. And so if you think about this, when you drop oil into a glass of water, you see little water droplets, and that's because they are forming this mycel, where the heads are pointing out and the tails are pointing in. Lastly, we're going to talk about proteins. Proteins are the most abundant organic compound in the human body. All proteins contain carbon, hydrogen, oxygen, and then one extra atom that we haven't talked about yet, which is nitrogen. Proteins are the most complex organic compound. Proteins have a lot of different functions. Now, amino acids all look the same. They have a central carbon, and off of that central carbon they have an amino group. The amino group contains a nitrogen. They also have a carboxyl group. A carboxyl group is a carbon with a double bond to oxygen and a single bond to a hydroxide ion. And then there's also the R group, and the R group is going to determine what type of amino acid it is. There are 20 different types of amino acids. Amino acids can bind together through the process of dehydration synthesis and form a bond between 2 amino acids that's called a peptide bond. And so when we have 2 amino acids bound together, that would be called a dipeptide, di for 2. And again, the process of dehydration requires removing water. Removing water and synthesis is to form something larger. So we can take 2 amino acids like glycine and alanine, remove water from them, meaning remove a hydroxide from one and a hydrogen from another, to form a larger structure, which is called a dipeptide. If we have a structure that's larger than a dipeptide, we would then call it a polypeptide. So through the process of dehydration synthesis, we could have form with a peptide bond another amino acid and another amino acid, and then finally we'd have a polypeptide. Now a protein is when we have a polypeptide of 100 amino acids or more. Now we have about 30,000 genes in our body, so we end up having about 30,000 different proteins in our body. Proteins can hold different shapes. A primary structure of a protein is just a straight linear chain of amino acids that are all held together by peptide bonds, and this is called a primary structure. So here's where we see the primary structure. Secondary structure results from bonds between atoms at different parts of the polypeptide bond, and they're held together by hydrogen bonds. So this hydrogen bonding can help to form either a alpha helix, in which we see that it's just in a helical type formation, or in pleated sheets, which we see over here, where there's almost sheets of chains of amino acids, all being held together by these hydrogen bonds. The tertiary structure is more complex, and there's a lot of coiling and folding of these helical structures. It starts to take on more of a 3D shape. And the last shape is called a quaternary structure, and the quaternary structure is where there is interaction between individual polypeptide chains to form a very complex protein molecule. 2 examples of this would be hemoglobin, and hemoglobin is the protein that will carry oxygen, and then also keratin. And keratin is protein that's found in the hair and the nails. It's a very fibrous protein. Proteins can also act as enzyme. An enzyme has 2 active sites where substrates can bind to them, and then the enzyme substrate complex will hold those 2 substrates together and facilitate the interaction between those two substrates until we end up with a bond between them to form a product, and then the product is released and the enzyme is left to be able to do the same thing with two more substrates.